So maybe we can start quite properly on time. So welcome back, everybody, to the Ketamine International Journal Club. It's our pleasure today to have Dr. Lauren Grock and Julien Dupuis presenting their work. They also kindly agreed to tell us a bit about themselves before actually digging into the paper. So, without further ado, I'll just pass the baton over to them.
Thank you. I can start. Thank you, Sarah. Thank you to all of your colleagues for the invitation. So I'm Laurent Grock. I'm a neurobiologist. I'm a research director for the CNRS in France, and I'm heading a lab working on the physiopathology of the NMDA receptor and synapse in general. And since a couple of months now, I'm the director of the Interdisciplinary Institute for Neuroscience here in Bordeaux.
And I'm Julien Dupuis, so I'm also a neurobiologist. I'm not working for the CNRS, I'm working for the French National Institute of Health, but in the same group as Laurent, so headed by Laurent. So we have these typical French research groups with several researchers. So we are actually 3 researchers working together in the group.
Should we start?
Yes, please.
Yeah, okay. So I will actually start the presentation, and Julien will actually mainly talk about the ketamine effect that we recently uncovered. But before that, I just wanted to give you a bit of a background of what we have been doing in investigating the regulation of an NMDA receptor, which I think is close to our heart for most of us, because of course ketamine is one of the antagonists of this NMDA receptor. And what I mean by that, that we are all very clear that the NMDA receptor is an absolutely essential receptor in the brain for glutamate synapse in particular, very important for many physiological regulation function of synapses, including the very well-known synaptic long-term plasticity that has been uncovered now many decades ago. And the NMDA receptor is also very important for the synaptogenesis, maturation, and many other functions. And not that surprisingly, a dysfunction of the NMDA receptor has been proposed to be instrumental in several neurological as well as psychiatric disorders. And in particular, of course, psychosis, but many other in which actually either an increased activation or lower function of the NMDA receptor has been proposed. We, the lab over the past decade, has been actually looking at how the NMDA receptor is actually trafficked from its synthesis to its synaptic localization. And we really focus our attention not on the synthesis of the receptor, not on the intracellular traffic of the receptor, but what happened to the receptor once it is embedded at the plasma membrane. And what you can see on the upper right here, this little movie, which is actually, yeah, nice, moving forward better.
Sorry, Laurent, we're not able to see your screen.
Okay.
We are seeing you, but not your screen, and I think you're trying to share it.
I can see it actually. Okay.
So maybe. Oh no, also the others maybe can see it. So.
Okay.
Sorry, maybe it's me.
Do you see it then?
We're
okay. So that's
okay.
Fine. Sorry about that.
No, fine. Don't hesitate. So actually the movie is really bizarre. But what you can see on the movie with this little green dot is actually the movement of a single NMDA receptor that is present at the surface of a neuron. And that's actually what is happening into your brain right now. This is not an accelerated movie. This is actually a video rate acquisition, which actually really changed our understanding of the way that receptor, not only the NMDA receptor, but actually all neurotransmitter receptors actually traffic once they are at the surface of the neurons. To make it short, receptor move like crazy at the surface of a cell. It's a thermodynamic process. Nothing else, temperature and movement of the membrane. And once they reach the synapse, and I will actually use a pointer if I can. but if not, that's no problem. But what you can see on the screen and on the graph is that once a receptor arrives into the synapse, they're actually trapped by many other proteins and mechanisms that retain receptor within the plasma membrane and within the synapse in particular. We uncover, and I will not enter at all into the detail on various mechanisms that are very important to regulate the movement of this receptor. Imagine that the receptor move quite a lot outside synapses and then being trapped into synapses. And when we started this journey kind of 15 years ago almost, or 15, 20 years ago, we were actually really interested by looking at intracellular proteins that was supposed to be at the time the regulator of the retention of the receptor and the regulation of the movement of the receptor. As you can see, things didn't go that way at all, in a way that now we really uncover that both extracellular molecules, it could be hormones, sexual hormones, stress hormones, amino acids, coagonists, many other proteins of the extracellular environment. Transmembrane interactions between receptors as well as intracellular proteins are highly regulating the movement of the receptor. So which really pushed us now to a vision, which is that the receptor, and the NMDA receptor in particular, is actually sensing its whole environment outside, in the membrane and inside the cell. And this environment is either dictating to the receptor, you stay or you go. And that's actually how you regulate the localization of receptor within synapses or outside synapses. And not that surprisingly as well, in that respect, we found that the fact that receptor move is not only important to regulate the amount of receptor that you find to synapses, but in addition, it plays an instrumental role in synaptic plasticity. And what I mean by that, if you can just look at the little movie on the upper left of the screen, you see in green spot that are synapses, glutamate synapses, and the red spot is a single glutamate-2B NMDA receptor that you can see jingling and moving at the surface of a cell. And when we use KMLTP, which is a synaptic long-term potentiation, we actually uncover quite surprisingly that the receptor was kind of moving in and out of synapses for a given period of time. And actually what we found was that this movement of the NMDA receptor is actually essential, required for a long-term synaptic plasticity. So it is not that the NMDA receptor that of course flux calcium, which is very important for synaptic long-term potentiation, as you can see here on this blue window with the inotropic function. But we uncovered that in addition, in the same time, NMDA receptor, the subfraction of the NMDA receptor, need to be rearranged and move around synapses in order to allow protein reorganization and intracellular signaling. So in order to have an NMDA receptor synaptic plasticity, you need to have, of course, calcium that flux in, but in addition, you need to have an NMDA receptor that are free to move and free to be reorganized. If you freeze only the movement of the NMDA receptor, you absolutely completely abrogate long-term synaptic plasticity, even if calcium is fluxing in. So the obvious question that we had was that then if it is that important for basic physiological processes, is that possible that NMDA receptor surface dynamic is also altered in models of brain diseases? And I will not go through what we did. I just want to emphasize one aspect which will be important for the investigation that we had with ketamine in particular. And for that, we use a model that I guess most of you are very familiar with, which is the anti-NMDA receptor encephalitis. It is a disorder in which patients express an autoantibody against the NMDA receptor, an extracellular epitope, and patient present first psychiatric symptoms, then neurological complications, and following immunotherapies, then you have a recovery for most of the patients. And all of the effect is mainly due to an autoantibody against the NMDA receptor. So what we and many other were looking many years ago was whether the autoantibody was an antagonist of the NMDA receptor, and many labs use various approaches to try to test whether antibodies were blocking NMDA receptor. Long story short, the autoantibody did not block or prevent a flux of ion through the NMDA receptor. So we were really facing this question at the time that, well, if the NMDA receptor autoantibodies are not acting at all on the inotropic function of the NMDA receptor, then who cares? That's most likely a kind of a side effect. And just based on what I just showed you a minute ago, then we investigated the putative role of autoantibodies on the movement of an NMDA receptor. And on the upper right, on the upper movie, you can see again a synapse in white, and you have a single NMDA receptor in green. This is again video rate, so most likely things happening into your brain right now. And when we expose hippocampal networks to the autoantibodies, but either from a healthy positive or just a healthy subject, basically nothing did happen. But the surprise came when we expose those hippocampus with patient autoantibodies, either patient with encephalitis or patient diagnosed with schizophrenia. Then in few minutes, we actually uncover a major reorganization of the NMDA receptor. They are still at the surface. They can still be activated, no doubt, but it's simply that the receptor are not located enough into synapses, and they are basically completely disorganized at the surface of the cell. And I won't enter into the detail, but what we recently even showed was that once the autoantibodies start to disorganize synaptic NMDA receptor, extrasynaptic NMDA receptor, that start to completely change the way they traffic at the surface of a cell, then over time, in the hour range, we see that there is a contamination of this molecular reorganization that move from the NMDA receptor to other type of receptors, including the GABA-A receptor. So over time, when you start to disorganize membrane NMDA receptor with those autoantibodies, then this effect, like a tsunami, kind of molecularly disorganize neighboring receptors that basically actually go towards the GABA synapses. And this have a major impact on the excitatory inhibitory synaptic drive, changing the overall hippocampal network activity. So obviously, from that point of view, we're like, is there a way for us to envision that that the NMDA receptor movement and not the activity of the receptor is actually a way for us to stabilize and to try to alleviate some of the effects that we observe in this situation? And this is where ketamine jump in, and I will let Julien tell you about it.
So can you see the pointer on the screen right now?
Yes, thank you.
Yes, thank you. Okay, so as Laurent was saying, so at this point, we were wondering whether drugs that are known to target NMDA receptors could potentially alleviate for the hypofunctions that are observed typically in the anti-NMDA encephalitis context. And so we started screening for different molecules that are either used clinically or in the lab to modulate NMDA receptor activity. And so we compared different families of NMDA receptor antagonists. The first one, and the one that is of main interest to you tonight, is the open channel blocker family, which comprises ketamine, also diacylpine or MK-801, and memantine. Those three ones, they bind inside the pore of the channel, close to where magnesium binds physiologically. And we compared the actions of those drugs with, on one side, competitive antagonists, so competing with glutamate for the binding to the agonist sites, so typically the AP5 or CPP, and also glycine site antagonists, so typically quinurenic acid or 7-CK, which will compete with glycine and D-serine for the binding to the coagonist site and GLIN1 subunits of the receptors. So the very first thing we did was to check whether all of these molecules efficiently silenced NMDA-mediated transients in the preparation that we're using in these studies, which is essentially dissociated primary hippocampal neurons. And what you can see here is we triggered on these cultures NMDA-mediated transients. So this is calcium imaging that you're seeing here, using applications of 5 micromolar NMDA either alone or in combination with the antagonist, so DAP5, ketamine, and K2-1 memantine at 51, 20, and 1 micromolar concentrations respectively. And what you can see is that with all of these drugs, you can efficiently silence NMDA-mediated transits. So from that point on, what we first try to do is to see whether those drugs had an impact on the trapping of NMDA receptors at synapses. And for this, we went back to one of the core techniques we're using in the lab, single molecule imaging. We basically labeled the receptors with quantum dots, so nanoparticles, and followed the movements of single molecules at the surface of neurons, so before and after a co-application of 5 minutes of an NMDA together with the antagonists in the presence of TTX to neutralize neuronal firing. And what we observe is the following. So here on the left, you have a receptor in buffer situation. So the receptor is labeled in red. You have a synapse labeled in green, and you see that it's entering, getting transiently trapped inside the synapse, and then released. What you can see on the right of the screen, I don't know if the movie is still playing. Well, what you should be seeing, at least, is a receptor that is much more trapped at the synapse than it is in the buffer condition after exposure to ketamine. We can try, we can try playing it again, maybe. So yeah, buffer condition on the left, and you should see ketamine condition on the right with the receptor. Yeah, well, obviously it's not playing really well, but the receptors are getting trapped much more after ketamine exposure of the neurons. So we can quantify this. So here on the left, you have representative trajectories in the different experimental conditions. And on the right, you have what we call MSD versus lag time plot. So what is represented is the mean square displacement, or MSD. So basically the surface explored as a function of lag time. And what you see is that before and after exposure to DAP5, it's the case also for quinurenic acid, you don't see much of a change in the behavior of the receptors at synapses. Now, if you expose them to ketamine or MK-801, and to a lesser extent to memantine, you see that you have a decrease in this MSD, which is consistent with an enhanced trapping, so less trafficking of the receptors at synapses, which are more stable. And this is also visible here on this graph where we quantified the synaptic residency time of the receptors, and you see that it's increased when receptors are exposed to open channel blockers. So open channel blockers have this property of enhancing the synaptic trapping of receptors at synapses. So the big question was, what's the mechanism that allows this? And if we go back to what Laurent described a few minutes ago, so there are 3 main families of actors that can regulate the trapping of receptors at synapses. So Laurent mentioned extracellular actors. So typically agonists and co-agonists of the receptor will modulate the residency time of receptors at synapses. And then you have protein-protein interactions, either with transmembrane actors, so typically receptors, adhesion molecules like FRNB2 receptors, here represented in yellow, also interactions with GPCRs. And mainly, and this is probably the main family of protein-protein interactions involved in the trapping of receptors, you have intracellular actors, and typically interactions with PDZ-domain-containing scaffolds, like PSD95, represented here in purple. And also physical interactions, but also post-translational modifications by enzymes, by kinases, such as CAM kinase 2 or protein kinase C, for example. So to make it short, we went for transmembrane actors. None of them seemed to be affected upon ketamine exposure. What I'll show you is the conclusions we came to, which points more towards intracellular protein-protein interactions. So how did we come to this conclusion? So to investigate whether ketamine had an action on the intracellular domains of the receptors that interact with these cytosolic proteins, we used Fret film imaging. So we used a Fret couple that was developed by the lab of Paul de Coninck, which consists in grafting a GFP fluorophore and an M-cherry fluorophore on 2 C-terminal tails of GLN1 subunits of the receptor. And so what happens is if we illuminate these fluorophores with blue light, we will excite GFP and trigger green light emission by GFP. Now, if the 2 fluorophores are close enough, so which means less than 10 nanometers in distance, you will start to observe energy transfer upon GFP excitation to M-cherry and the appearance of red light emission by M-cherry. And this will coincide with a decrease in the lifetime of GFP. So basically what you can do then is you can monitor this lifetime of GFP as a proxy for changes in the conformation of the receptor. And that's what we did before and after exposure to the different drugs we mentioned just before. So here, color-coded, you have the lifetime of GFP. You can see that the more you go to the blue, the shorter the lifetime is, the more you go to red, the other way around. And what we saw is that basically exposure of the receptors to DAP5 did not change much the lifetime of GFP, so the conformation of these C-terminal domains. Same with memantine. Now, if you take ketamine and MK-801, you see that in both cases, you have a decrease in this GFP lifetime, which is consistent with an increase in FRET efficiency. So in other words, those two domains are coming closer. So there's a change in the conformation of the receptors. So just to mention, this is a lightened version of the figures that you find in the papers. So for the sake of clarity, I didn't represent necessarily all of the experimental conditions. Okay, so change in conformation. Now, how does this translate to change in protein-protein interactions? So to answer that question, we use FRET again, this time not intramolecular FRET within the receptor complex, but between the receptors and PDZ domain-containing proteins, and we took PSD95 as a prototypical PSD protein. So PSD95 was engineered to incorporate the DM-cherry fluorophore, and this time we're using lifetime of GFP fluorescence as a proxy for protein-protein interactions. This is also a FRET couple that was developed by the lab of Paul de Coninck, and what we first observed is a reproduction of something that Paul had published, which is that if you activate the receptor with an application of NMDA, you see an increase in GFP lifetime, which is consistent with a dissociation of the complex. Now, if you apply DAP5, together with NMDA, you see that you can prevent this decrease in this increase in GFP lifetime, so this decrease in FRET efficiency. And what you can see actually is that application of ketamine and MK-801 increases even more this lifetime of GFP. So in other words, it strengthens the interaction between NMDA receptors and PDZ domain-containing proteins. So this could be an explanation why receptors get more trapped at synapses upon ketamine binding. So to finally confirm that, we went back to single molecule imaging and monitored the behavior of either wild type or mutant receptors incorporating point mutations in the C-terminal domains of GluN2A and GluN2B subunits, which interact with the PDZ domains of scaffolding proteins. And what you can see is that while wild type receptors are slowed down at synapses and see an increase in residency times upon exposure to ketamine, mutant receptors do not retain these properties. So in other words, this enhancement of receptor trapping at synapses depends directly on interaction with scaffolding proteins of the postsynaptic density. Now, does this property of ketamine have anything to do with its clinical action? So to try and answer that question, and I'll bridge back onto what Laurent described in his introduction, we went back to anti-NMDA receptor encephalitis. So if you remember, patients with encephalitis express these autoantibodies targeting extracellular domain of the receptors, and by binding these receptors, they disorganize and they destabilize these receptors from synapses. So what we did is we went back to single molecule imaging, monitored the behavior of receptors after exposure either to IgGs from healthy individuals, from patients with encephalitis, either alone or in combination with ketamine. And what you can see here is that while patient IgGs triggered an increase in the area explored by the receptors at synapses, so they destabilize receptors from synapses, their co-application together with ketamine actually restored proper trapping of the receptors at synapses. And by doing so, it also restored the amounts of receptors found at synapses. So patient IgGs triggered a progressive depletion in the amounts of receptors at synapses. So this is immunocytochemistry images that you can see here. And this decrease in NMDA receptor content at synapses was also compensated by ketamine. So at this point, you may tell me, okay, very well. So by doing this, ketamine somewhat compensates the depletion in receptors at synapses, but now you're left with receptors that are inotropically silenced. So what's the point of that? They're still unable to signal. And so we try to see whether actually some signaling at least would be restored by this action of ketamine. And for this, we went back to FRET imaging using a reporter of calcium calmodulin modulated kinase 2, so downstream kinase directly associated with an NMDA receptor. And this reporter consists of a GFP and a REACH, so red light emitting fluorophore, grafted on two ends of the catalytic site of CAMK2. So you can monitor directly a CAMK2 activation by monitoring the lifetime of GFP. So when the kinase gets activated, the catalytic site opens, so both fluorophores get separated. So there's less threats, and the GFP lifetime should increase. And that's exactly what you see in this control condition. So here, hippocampal neurons expressing this CAMK2 reporter were exposed to IgGs from healthy individuals. You see that if you apply glutamate on top of the neuron, you see an increase in GFP lifetime, which is consistent with CAM kinase 2 activation. So that's what you can see also on this graph, monitoring evolution of the lifetime as a time lapse. Now, if you do the same after exposing neurons to patient IgGs, you see that there's a marked decrease in the amplitude of CAMK2 activation. And actually, co-application of ketamine together with the IgGs restores the full extent of CAMK2 activation. So not only does ketamine restore the trapping and the amount of receptors at synapses, but it also restores at least part of receptor signaling. So now the final points that we wanted to make, and this will be my last data slide, was whether actually this would translate in vivo and would allow also compensation for some of the deficits associated with these antibodies. And to try and answer that question, we actually implanted animals with osmotic pumps and cannulas directly dipping into the ventricles to actually allow chronic infusion either of antibodies alone or in combination with ketamine. So we did this for a couple of weeks and then submitted the animals to a battery of tests. So I won't go into the details of the full battery, but just the main features that we observed, the main dimensions that were affected by the antibodies were typically anxiety and depression-related manifestations. So you can see here, for example, in the sucrose preference test and full swim test, that there was clear anhedonia and despair in the animals exposed to patient antibodies. And we also monitored for sensory motor gating deficits using the pre-pulse inhibition of the Stottle test. And you can see that this is also affected by exposure to patient IgGs. And in all three cases, you see that actually ketamine is able to fully restore the behavior of the animals and to compensate for behavioral deficits, both anxiety and depression related and sensory motor gating deficits associated with exposure to patient antibodies. So just to wrap this up and open a few perspectives that we can discuss. Our conclusions from this study is that ketamine has this very unexpected action of enhancing receptor trapping by favoring interactions between NMDA receptors and PDZ scaffolds. And this may be of interest clinically speaking, because this trapping prevents molecular and behavioral deficit caused by patient antibodies. So typically situations of an NMDA receptor hypofunction could paradoxically be improved by this action of ketamine. And now, yeah, in terms of perspectives, there are at least 3 points that we'd be very happy to discuss. So first, there's this question of how much ketamine could be used as a treatment in anti-NMDA receptor encephalitis. So which patients would it be of interest for, and at what time point of the disease should ketamine be administered? So that's one first thing. Also, and more related to the compensation of anxiety and depression-related deficits, are NMDA receptor synaptic reorganizations a more general mechanism contributing to ketamine's antidepressant action? So that's also something that we may discuss. And more generally, how much targeting NMDA receptor surface trafficking and synaptic trapping may be a new target of interest for therapeutic intervention. I'll stop here. Just mention, I know that we're not going into details of the acknowledgements, but this is just the people in the lab who have been working on this project, and here collaborators, and especially clinician collaborators, we're working on these projects hand in hand with, and the fundings. Thank you for your attention.
Thank you very much. Very thorough presentation. I wonder if anybody has any question already. Feel free to just ask the question directly, or if you're not comfortable, you can just write them in the chat and we can read them out for you.
Should we stop sharing the screen, or you want to keep it?
I think it's fine if you want to stop sharing it.
Yeah.
And then eventually, if there is any specific question and you find it helpful to go back to your graph, you can start with sharing. Well, Rupert and she raised their hand.
I just want to say that is just amazing. That's just incredible. So thank you so much. It's just exhilarating to hear just the sort of argument and the science. So thank you very much for.
Thank you so much
for that. I know this is tangential, and I'm sorry that it's going to be tangential because I'm a clinician. I had a patient who had NMDA encephalitis who responded to ECT. And I'm just wondering whether that's worth thinking about with your next phase with the animal models, perhaps. I don't know what ECT does to this trafficking, but it was very impressive. It saved his life, and then the next time he got it, he didn't get the ECT and he died. It was a very sad story. But it's certainly, you know, as a ketamine clinician, I sort of can't, you know, I'm terribly simplistic, so you have to forgive me for that. But I tend to think, you know, if something responds to ECT, it might respond to ketamine. It obviously, I mean, but when I saw your paper and I sent it to Belinda Lennox, who's our sort of encephalitis or NMDA encephalitis guru, she, like me, with her first reaction was, "What, you mean give ketamine to people with psychosis?" That's going to be a tricky one. But I'm much less fearful than she is, because I think ketamine's a very safe drug, and probably even in psychosis, particularly if you've got people who are, you know, in extremis. So to answer your question about dose, what dose would you use? I mean, we know that ketamine at 0.5 milligrams per kilogram works in depression. It's a safe dose. You'd probably start there, wouldn't you? But you might need a higher dose. So those are some comments. And I have another comment that I want to make about your very first slide, which was about corticosteroid and exogenous influences on this, which of course are sort of, in a way, potential biomarkers of response. And so, but maybe I don't know if you want to respond to that first set of rather divergent sort of speculations.
Yeah, I can start. And first, thanks a lot for your comment. I would actually love to have just a few answer on your very important question, which unfortunately I don't. Regarding ECT, I would actually love to understand and to test whether ECT is acting on the specific receptor present at the plasma membrane of a neuron. I have no idea. I don't think anyone knows that because I'm not aware of any experiment done this way. But I would kind of tell you that each time we and others have been playing with the overall activity of a network, and it was very much done in a way to try to modulate synaptic transmission, either improving or increasing, decreasing transmission. In most of the cases, actually the movement of receptor, the plasma membrane, were regulated up or down, depending on the way that you stimulate and so on. So I won't put my hand into the fire, but I would maybe put a finger saying that I think that ACT or such type of a trend most likely have a profound effect on the way that the receptor move. And once I say that receptor move, you may tell me, well, I mean, receptor move all the time. What's the point? But you have to think that it's a whole an equilibrium. because most of the receptors are outside synapses, and they diffuse, they enter synapses, they spend few minutes at most into the synapse, and then they leave the synapse. Another ones come. So you have a balance. So if you start to change the way receptor outside synapses move, more or less, you will completely change the equilibrium, and you will completely change the amount of receptor into synapse. It's very much biochemically. It can be re-explained on a very simple biochemical means. So I think for ECT, that's most likely a possibility that you have this regulation. Back on ketamine, what is the dose? I think everything is here. Everything is in the dose. I have no idea what will be the effect on patient. I think with encephalitis, the whole point is when. Because what we just show here in this animal model is that if you are facing a situation in which NMDA receptors are pushed away or are not stabilized anymore into the synapse by some ways, then ketamine is an extremely powerful way to retrain and keep receptor within synapses. Even though they are locked, iotropically speaking, they are retained into the synapse and most likely have other type of way. So when is it appropriate for patients? What is the dose? A very low dose have the effect that we observe in vitro and in vivo. Of course, we have to be very careful, but last hours. So that's really a long-lasting effect of the molecule that is observed there. How much this translates that to patient? That's something that now we want to really push forward with some specific trial in some specific condition. Because again, we have this idea that if you have an hyperfunction of the NMDA receptor at a given time, ketamine may paradoxically be rather good to retain and keep some NMDA receptor within synapses. Want to?
No.
Thank you. Thank you very much. I'm going to ask my other question now.
I think there were a few questions in the chat as well.
That's all right. Yeah.
saw a few ones popping up.
Could I go for your very first slide? Sorry to hog it. If you could share your screen and go back to your very first slide.
Yep.
So if you share your screen.
Yep, I do that. There you go.
I'm interested in the idea that corticosterone or steroid. Actually, no, it's the next one. Sorry.
Yeah.
Yeah, it was this one, right?
Yeah. That corticosterone actually sort of reduces the NMDA at the synapse. Have I got that right? Is that what this means, or it's reducing the diffusion of the?
It decreases the diffusion of the receptor, and
so it's a bit like ketamine in some way.
Yeah. So in some way, because with corticosterone, something that actually we don't know much for ketamine, but clearly here we have a difference with different subtype of the NMDA receptor, glutamate containing, glutamate containing for corticosterone. And a very striking effect of corticosterone is that everything that we describe is non-genomic. So the effect of corticosterone, exactly like ketamine, happen in the first seconds, minutes. And that's, I think, something that's still completely, to be absolutely transparent with you, still puzzles me really deeply, is that all of these molecules are really potent regulator of the movement of the receptor, whether they like to stay in synapse and so on, and those effects are really membrane dependent. You don't have any genomic regulation. You don't have most of the process that are, of course, important for physiology and pathology, but in that particular cases, those molecules have a way to regulate the way that receptors move at the plasma membrane. And that's the same for ketamine. As Julia mentioned, you put ketamine and you see the effect in the seconds. That doesn't take much. So basically, it's a very much a molecular cascade, such as corticosterone. Same was observed with estrogen, for instance, as well.
Maybe to complete on Laurent's answer, this is essentially an acute action of corticosteroids. The one thing that we don't know, but we're investigating this right now, is how much a prolonged exposure to corticosteroids actually has the same type of effect, or if it triggers something different. Because obviously, prolonged application is not necessarily going to engage the same type of glucocorticoid receptors. So the picture could be different upon chronic stress, for example, and chronic high levels of corticosteroids. This is something we still don't know.
I saw a presentation recently that suggested that actually almost the only predictor of treatment response to ketamine was high corticosteroid levels, cortisol or ACTH levels. I don't quite know how that all fits in with that, but anyway, that's for another day.
Yeah, but that is, as Julien mentioned, I think here we are in a more chronic situation where then corticosteroids have other type of effect on the NMDA receptor and the AMPA receptor and many other at the surface. So that's what we are investigating. It's absolutely not a flatline effect. We clearly have a multiphasic effect of stress hormones, sexual hormones, and here what you have on the screen is very much the very rapid acute effect. And if I may, because I went really rapidly on that, I mean, we were very, very surprised. I mean, 15 years ago, I would have told you that this type of graph is just impossible. We and many others were looking at the D protein, deregulator of the movement of receptor, where an NMDA receptor should stay into the synapse or leave the synapse. We were really looking for that. And actually, more we dig, more you find as potent regulators from the physiological system, for interleukins, inflammation. I mean, you have all of those that are strong regulators of that. So I think we have a very wrong thinking, which is that we are looking at the regulator of the movement of the receptor. Actually, the receptor, and the NMDA receptor in particular, is actually an amazing molecular sensor. And basically is sensing this whole environment from the top of his head to his toes. And that's basically integrate and say, go, run, or stay. And that's extremely powerful for a neuron. It's all thermodynamic. It doesn't consume any energy. It's just your temperature that make the receptor move or be regulated. So I think now it's a complete new conceptual framework in which we and many others are really trying to put that together, because the addition of each of these molecules by itself is very difficult to integrate, but maybe because we just don't have the right software to think about that in your brain, all of these molecules, they're actually all together. And the receptor is actually exposed to all of them. Different concentration, of course, but all present. And it's all a question about integrating those signals into organization.
Thank you so much.
I think we have a couple of questions from the chat. So we have Chang.
Do you want me to read those?
Sure. Yeah,
so I can take the first, and then we can move on. Help me understand in simple way. In disease, we don't have much NMDA receptor traveling the membrane. Is that it? Ketamine made them travel more. It's for ketamine in that particular case, it's the opposite. That in the situation with autoantibodies against the NMDA receptor, they actually tend to be disorganized, so they move more because some of the interactions are actually impaired. So ketamine has a is very powerful in stabilizing receptor within synapses. What we didn't show you is that there are other models of brain diseases in which NMDA receptor are locked on the pathological way into synapses. And then in that particular case, what would be very interesting to do would actually to increase the movement so they can go back to an equilibrium. So it's really like a U-shaped curve that we are thinking of for the future, which is that you may have brain condition in which the receptor are completely disorganized, just running all over the membrane and not stabilizing to synapses. And then ketamine may do a good job because it may help stabilizing. But you may have a situation in which receptors are frozen at the plasma membrane. In that particular case, you will actually like to increase the movement of the receptor. So I don't want to say that there's one path, which I don't believe a second. There is one path, one regulators. I think it's just a balance. Too few, too high movements can be detrimental. You take the other one?
Yeah. The second question was, how long is the effect of ketamine on NMDA receptor entrapment? Well, that's a good question. We don't really know. I would say in vitro, it lasts at least as long as the drug is bound to the receptor, and it all depends on the off rates of the drug. So typically the action, the effect of MK801 is pretty stable across time. I mean, the off rate is so low that MK801 traps the receptor at synapses for very long periods of time. So ketamine will dissociate faster. And you can actually boost that. So that's something that was actually used in Helen Hu's last paper. So where she triggered activity in the lateral habenula to force dissociation of ketamine from the receptors. So, but I mean, otherwise it's pretty stable. It can last for at least several tens of minutes.
Then ketamine is categorized.
Yes, Sarah?
Sorry, 1 second, if I can piggyback on that question, right? I think one of Helen Hu's recent paper was exactly about the trapping on the ketamine at the level of the lateral habenula to explain a bit not only the rapid effect of ketamine that we see as an antidepressant, but also the sustained. So this was one of my questions. Do you think that this mechanism may help us understand a bit both, like the rapid and the sustained effect of ketamine, or does it talk more to one or the other?
Well, I think what Helen Hu meant with trapping and what we mean with trapping is not the same thing. So by trapping, she meant trapping of the drug inside the pore of the channel, of the receptor. What we mean here is synaptic trapping, so trapping of the receptors inside synapses, which is not something that she investigated. But I think, yeah, what she means is that in the lateral habenula, where activity is really high in depression, so most likely ketamine will preferentially act on structures where activity is super high, like the habenula. And once it has acted there, if activity is downregulated, it will remain trapped there for a long time, which I guess is the what she's putting forward as an explanation for the sustained action of the drug. Then, of course, with the downstream actions on the cascade of downstream action on the connected structures linked to the habenula. But yeah, what we mean here by trapping is more the trapping of the receptors at synapses. I don't know if this answers your question in any way, but
it's fine. Thank you.
Is it responsible for the sustained action of ketamine in depression? Well, honestly, I don't think we can answer that question so far. That's the reason why we listed it. I mean, we're obviously working on it intensely, trying to figure out how much these mechanisms also contribute to the antidepressant action of the drug. But so far, I mean, I can't give you any answer on that. I don't know.
And just to speculate a bit further on that question, if we compare to Aileen work, what we don't know from our side, all of the movie that you saw with the movement of receptor, NMDA receptor, all of the other, is mainly based in the hippocampus, hippocampal network, a bit of a cortex, that's it. So one of the key questions in the field today is how much the exact same NMDA receptor move at the surface of a neuron in the hippocampus, in the cortex, in the striatum, in the lateral habenula, whatever you can name all of the structure. And no one has a clue. So is the brain a mosaic of very different type of movement because of, you saw all of the molecule can actually regulate that. So if you change some of the molecule present in the surrounding or within the neurons, you can actually regulate that. So that's one of the questions that is really on the table for several labs, which is to really map the brain and to test how much certain brain areas are very favorable for the movement of receptor, maybe some are on the other way around. So that's an open question.
Thank you.
So is ketamine is categorized as an NMDA receptor antagonist. I am curious to conceptualize how it restores normal NMDA receptor function. That's $1 million euro, pounds, dollar question. That I think we are extremely interested in that, because as I guess you will think of, if the NMDA receptor is blocked, inotropically speaking, then what's the point? I mean, then basically it is muted. So we all learn that then nothing is happening. Well, I mean, we don't know. we another, as you may be aware, have really been digging into this non-inotropic function of the NMDA receptor. And the NMDA receptor is actually bringing a lot of molecules with it. Actually, we don't know with the egg and chicken, but basically the receptor can serve as scaffold for chem kinase 2 and many other kinases. When the receptor moves, we clearly see that other molecules start to move, which what we call a shuttle effect. That if I move, I don't move alone. So most likely there are other functions of the NMDA receptor that are as important as the inotropic flux of calcium in that particular cases. I am an electrophysiologist, so I learned when I was doing experiment whether you have APV, AP5, or MK1 or ketamine, where it was mainly KET1. And when you want to block an NMDA receptor, who cares? I mean, you just block the current, and it's very efficient. And then, but actually now that we all go back into the literature, we actually realize that these molecules have a very different effect on some more kind of complex phenomenon. So I think this may be just the tip of the iceberg, that there is non-inotropic regulations, non-inotropic cascade that we basically don't grasp very well. I mean, we just need to face the reality. And it's most likely protein-protein interactions, macrocomplexes that are very important, not only for the NMDA receptor, but for all of the proteins in the synapses as well. So hopefully in the next years, we and others will have better answer to this key question. But when we think about an NMDA receptor antagonist, just blocking the receptor, we automatically now look at what those molecules are doing on the traffic, and you will be very surprised actually to see your favorite antagonist doing very bizarre things on the receptor. Yeah. Or maybe
maybe this one is for you.
Yeah, without the disease where it is frozen. We had a study in which actually you increase inflammation, interleukin-1 receptor, in particular interleukin-6 TNF, where over time we clearly see that we have a massive reduction into the traffic of the NMDA receptor. So it depends on the time, depending on the cocktail of molecules, it can really go either way. And even for the autoantibodies, actually, what didn't we say here, that if you let for hours and even days, then you start to have a downregulation of the movement of the receptor. So it's not even like one mechanism that is observed there.
Yeah, then there was, is the problem with the world trapping two different phenomena? Yes, I think that's the point we discussed. Yeah, what Helen means with trapping is trapping of the drug inside the pore, and what we mean with trapping is trapping of the receptors at synapses. It's a pity that both papers were in the pipeline at the same time. Otherwise, maybe we would have phrased it differently. But yeah, two different meanings.
And what is I think also appearing over time now is that trying to understand how the receptor, and it's not only true for NMDA receptor, we could have almost given very similar presentation with the GABA-A, with the AMBA, with also, and with many GPCR as well. So I think there's kind of a new era that is really in front of us, which is for the NMDA receptor to really decode what is the mechanism that regulates the way the receptor like some environment more than others, and spend more time in some environment more than others. And if we now gather more and more evidence that this is important in some models of pathology, I think that would be a game change for us, but for the pharma industry as well, which have been starting to make this term, to really think not only about agonist antagonists, which we know all of the advantages and disadvantages for the NMDA receptor in particular, but to look for stabilizer. And I don't want to say trapper, because then it would be difficult, but stabilizer of the receptor into given places. And that may actually change quite a lot for NMDA receptor and some other. So we need to think about a good name, not a trapping, not a stabilizer neither, but something, because stabilizer look like a mood stabilizer, so maybe it's not good neither. But we have time to think about it.
So we had another question popping up. Any thoughts if the isomers of ketamine behave differently with the NMDA receptor? That's a very good question that we obviously have in mind. So we didn't test for that until now, essentially because of access problems to the enantiomers. So that's it's very difficult to get. We can get ketamine quite easily because we use it as an anesthetic for laboratory animals. But R-ketamine and S-ketamine are not that easy to actually get. What we did test though is the downstream products of ketamine, so the metabolites of ketamine. And typically what we can tell you is that the hydroxynorketamine byproducts, for example, don't have the same type of effect on an NMDA receptor at synapses. It's really only specific of ketamine directly. Whether it's stronger or weaker with R and S ketamine is something we don't know.
Okay, I think we are coming towards the end of our session together. So unless anybody has one last question to ask, I think we can conclude this here. Thank you so very much for the presentation and taking the time to answering all these questions. It was great. We are clearly looking forward to the next step and the next terminology on the field. And yeah, and thank you so much. And I guess I'll see you all again in a couple of weeks. We have Thomas Liebe from Martin Walter Group talking a bit effects of ketamine on the locus coeruleus and using 7-Tesla MRI. So thank you so very much.
Thank you very much.
Thank you.
Thank you.
For having us.
Thank you. Bye.
Amazing. Thank you.